We will now show you how to take a DNA sample. Please watch carefully before you take your first sample.
When you receive the kit, the best way to do the sampling is to open up the kits individually, and this avoids contamination. So for example, if the mother's going to do the swab first, she takes her swabs herself and puts them in the envelope, then do the child, and then do the father. And that means that there's no possible contamination between the different samples, and nobody else has touched the swabs apart from the person who's having their sample taken.
And also it helps if everyone is there at one time, so they have confidence in the results and don't think that anyone's been tampering with the swabs without them knowing.
Okay, so it's very easy. You take out the swabs, which are in a plastic envelope, open up the swabs, put them into your mouth, and just rub the inside of your cheek very, very gently. It doesn't have to, it won't hurt it in the slightest. And then once you've taken the swab, hold it in the air for 30 seconds just to allow the swab to dry, and then place it in the envelope.
Now we provide 2 swabs for each person, so you should take both swabs. This is because if the first swab doesn't work, then we need to test the second one. This is much easier to do if we have a second one there, rather than sending back to you to get a second swab taken.
It's very important to put the swabs straight back into the envelope and not on the table to avoid contamination. And it's also important to put the cotton wool tip head first into the envelope.
This means it's very, very easy for us to get the swab out without touching the bit with your DNA on. And also it means that there is no contamination. For example, if you put it down on the table, if you put it down on the floor, you could pick up DNA from another source. We don't want that to happen.
Each stage of the process is important because it's important to make sure, first of all, that you're not contaminating your swab. Secondly, that you're putting the correct swab in the correct envelope. And thirdly, that you've filled in all the details, because all these will affect the quality and the result of your test.
You need to be especially careful when taking a DNA sample from a child. Remember, it's cheek cells we need, not just saliva.
To take a sample from a child, it is first important to ensure that the child has not been recently breastfed if it's a baby, because then we can get contamination of the mother's DNA in the child's mouth. Other than that, it's a fairly simple process to just put the swab inside the child's mouth and just wipe on the inside of the cheek for a few seconds. That's usually enough to get some DNA.
Often it's an idea to show them taking the sample with mum first, and that gives them confidence and they can see how it's done. Then mum can take the sample from the child, or some children like to do it themselves, and just rub the swab on the inside of their cheek, and then you can show them putting it into the envelope, and they might even want to do the second swab themselves.